and ICF. So what are the ECF and ICF? First, let's talk about the ECF, extracellular fluid. Extracellular fluid is that fluid which is present outside of our cell body. Fine. So outside of the cell body, there are lymph. We have discussed a bit in the about the lymph in animal tissue part, right? Uh, we talk about the blood, and extracellular fluid is rich with the sodium and chloride. Fine, that is the property. These element, these ions or minerals, they have been asked in medical examination, right? So the ECF is rich with sodium chloride, potassium phosphate, potassium chloride, and all that. That combination. The second part is ICF, that is intracellular fluid. So from the name itself, we can say this is the fluid which is present inside our cell, right? Intracellular fluid. So basically, inside our cell, we know the thing which is in liquid form, or rather liquid form and gel format, that is protoplasm. So protoplasm is a part of ICF, and it is rich with the potassium and phosphate, right? Further, the blood make 30 to 35% of ECF. So what is the rest? Rest part is the lymph, right? So 30 to 35% part of the blood is ECF is blood. An adult person of 70 kilogram, it have 5.5 liter blood, 5.5 liter that blood with a pH value of 7.4. And from the pH itself, we can say that this is slightly basic in nature. Right, Shedika?
Yes, sir.
Now, what is the plasma? What is the blood? So from this example, we can understand. But you do not have notebook. So I'll just, you can take the screenshot of this one. Fine. First, understand.
Okay, sir.
Try to understand what I want to say. Look, uh, let's say this is, there is a tumbler or there is a jar where we fill the jar with the blood. This blood belong to a normal human. This will directly come in the contact with the oxygen in 6 to 7 minutes average in human being. Maximum can be 20 minutes. The blood will clot. What is the process of clotting? How does clot formation take place? We'll study in next slides. But here, just to let you know the component of blood, I want, that's why I am talking about the clotting over here. Here I am not focused on the clotting. Here I am focused the component of the blood, because when the clotting will take place, the component gets separated from blood. So let's keep this blood in open, right? After 6 to 7 minutes or a maximum in 20 minutes, the blood will change its from form from the liquid. There will be a liquid part, that is blood plasma, and there will be a clot like gel like structure, a clotting structure, that is called blood clot, right? So that this kind of the picture will appear in front of this blood plasma, or light yellow color liquid which gets separated. This is 55% part of the blood. So look, 55% part is purely liquid and 45% part is solid. When we talk about the blood plasma, so we call this blood plasma. Blood plasma is rich with, this is light yellow in color. This is made up of 90 to 92% water, right? This question have been asked in medical examination, right? It had been asked. So 90 to 92% part of the blood plasma is made up of purely water. Rest component of hormones are there. They remains dissolved in our blood. Carbohydrate means sugar is there. Protein is there, means amino acid. Fat is there, and there are minerals. Right. This is component of, these are the composition of the blood plasma. Now let's talk about the blood clot part. Means the fiber, this solid part. The solid part which gets separated from this blood or liquid part, it constitute 45% part of the blood. It is made up of RBC, red blood corpuscles, WBC, platelet, and there are two protein, thrombin protein and fibrin protein. So look, Shedika, what actually happen whenever suppose that we get injury, any external injury. Because of this external injury, what will happen? The blood will start coming out from the, or oozing out from that wounded part. When blood will start coming out of that part, what will happen? That cut part will be sealed by a gel-like structure, that is blood clot. So blood clot seal that, right? So basically, after sealing, their formation of a layer, dead layer will take place. The formation of dead layer takes place because of this thrombin protein and fibrin protein, right? These are the two solid protein which make a covering or healing part of the that wound, right? And they prevent the blood to come out of or excess loss of the blood. Did you got it, Shadika?
Yes, sir. Yes,
Take
sir.
this, take this screenshot.
Okay, sir.
So you don't have any pen, paper, and all that. Are you like somewhere else?
No, sir. No, sir. Baggage, the luggage is too heavy to carry any extra
Oh,
books, so
yeah,
I only got the course book.
okay, okay.
I'll
No
be
issue.
going out today and I'll get a book today.
No issue, no issue, no issue. You can take the screenshot. And
Done, sir.
we will, I'll dictate in the next class. So you have to note it down in the next class, as well, right? Because these
Yes,
all parts
sir.
are important. These all parts are
Yes,
important.
sir.
And you will get lots of questions from here in both normal exam and as well
Oh,
as,
okay. So it's important, this
yeah, it's
part
important.
specifically.
Clotting part is very important. You can get the question from the clotting. You can get the question from double circulation. You will get the question from WBC. There are a number
Yes,
of things which
sir.
are very important. Yeah, it's very important.
Okay, sir.
Look, when we talk about the plasma, we have already understood 55% of the blood is plasma. Out of this 55%, 90 to 92% part is of the plasma is water, right? Mineral, nutrient, and proteins are found. 6 to 8% protein of the plasma is albumin. Now, I have a question. Try to understand what exactly I want to ask. Look, Shadika, whatever you eat from morning till night, after you wake up and at the last time when you go for the bed, right? Between this duration, whatever you eat, that all are acidic. How?
They
We take
all are?
water, acidic.
Yes,
You know,
sir.
everything which tastes you good, everything, whatever we eat that tastes good for us, that is 100% that is acidic. Because basic things are bitter, they are not good in taste, right? And whatever the food we take, that is all tasty, most of it, right? So that is all acidic. So all acidic things are tasty, and basic things are bitter, and they are not that much tasty. Sometimes we have it, but very rarely. So from the morning to evening, whatever we eat, we drink the water. So what is the water? Water is slightly acidic. It is not neutral. The pH value of water, you know it or not? The pH value of water, what we drink, that is mineral water, that is not neutral. It's acidic.
It's five, around five.
5 is too much. No, no, slightly acidic. It may be something
Okay,
5
so
point something, right? 76 point something, right? So
yes,
you know
sir.
why? Whenever we suppose that I have this water bottle, right? As soon as I open the, suppose that this is sealed and water is inside, pure water is inside. As soon as I open the knob of this water, what will happen? The carbon dioxide will go outside and that will react with water and make carbonic acid. It becomes acidic, right? So that's how water is always acidic. The water we drink. We do
Okay,
not talk
sir.
about that distilled water which is found in the lab. That is neutral.
Yes, sir.
So water, that is acidic. Protein, amino acid, that is acidic. Fat, that is fatty acid, that is acidic. Sugar is acidic. These are the main component which we eat. So we eat everything acidic, but our blood is basic. How? By the our blood should be acidic. It should be or not. Because whatever we eat, finally, in the nutshell, it goes to the blood. Did you got my logic?
Yes,
Logic?
sir. Yes, sir.
How, Shedika, how it's possible? It should be acidic, but it's not acidic. Do you have any idea, any clue?
It gets neutralized by the other components in our body,
Yeah, exactly.
other bases and acids, which
Yeah,
help
you're close to the
with the
answer.
digestion.
Yeah, you're close to it. No, no. After the absorption in the blood, this is the part. This is the property of the blood that neutralizes, not the
Is
digestive
it
system.
the is it does it get neutralized by the carbon dioxide that we breathe in?
No, 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 not at all.
Oh,
No.
sorry, not breathe in, sorry. That's taken out from our body.
Look, carbon
No?
dioxide is itself carbon dioxide itself acidic. That is
I
itself
don't know,
acidic.
sir.
Look, that is a question and try to understand. This question I've been asked multiple time in different way in medical examination. Here I have written, if you go to through the plasma, it is made up of mineral, nutrient, and protein. 6 to 8% of protein of the plasma is albumin. Albumin is the same protein which is found in the egg white, right? Plus globulin protein. Globin protein, globin protein make hemoglobin. It combined with the prosthetic group iron, and it make the hemoglobin. But this albumin protein, it remains dissolved with our blood. It maintain the pH value of the blood. It do not allow blood to change into basic acidic. It keep the main role of albumin protein, which keep the blood basic in spite we eat each and everything that is acid, right? It work like a buffer solution. So what are the buffer solutions? Buffer solutions are those solutions which resist the change of the pH value of solution, right? So they work as a buffer solution. They do not allow the acidic thing to let the change the pH value of the blood. They maintain the blood pH value. Got it?
No, sir.
Look, every acidic thing get dissolved in our blood, right?
Yes,
Whatever
sir.
we eat, that get dissolved with our blood, absorbed by the blood. Now, when they try to change the pH value of the blood, right? Now come the role of the albumin. Albumin protein is already present in our blood, right? It resists the change of the pH value and it convert them into basic component.
Oh, okay.
So basically, this is the albumin protein which maintain the pH value basic, right?
Of
It
our
do
blood.
not allow the blood to get acidic because of whatever we have eaten, that is acid. Did you got that?
Yes, sir. Okay. Yes, sir.
So albumin is known as buffer solution. Albumin work as a buffer solution. Got it? So what are the buffer solution? What is the definition of buffer solution? Buffer solutions are those solutions which resist the change of the pH value of the blood. Sorry,
So
which resist
albumin is the only buffer. Uh,
exactly. Albumin work as a buffer solution. It work as a buffer solution or buffer salt.
okay. So there's other buffer solutions too.
Yeah, other buffer solutions, they are the basically buffer solutions are we use in the chemistry lab. So they are the
Okay.
solution which
And this is
maintain the
the one
pH value.
present in our body.
Got it? Yeah.
Albumin is the one
No, no,
present in
no.
our
Albumin,
body.
yeah,
Yes.
albumin work as a buffer solution in our body.
Yes, sir.
Got it?
Got it. Yes, sir.
Other thing which are present in the blood, lysozyme and properdin. Important. Properdin have been asked multiple times in medical. Lysozyme, this is an enzyme. Keep it in mind. It is found in tear. It is found in saliva. It is found in sweat. Right? It is found most like many part of the body. So lysozyme is an enzyme which kill the bacteria and germs. Properdin, it is a protein component. It act at antibiotic substance. Got it? So
Yes,
lysozyme
sir.
and properdin both are the antibiotic, means they kill the germs. One more thing which is present in our blood, which is responsible for which do not allow blood to clot normally. Look, when blood come in contact with the air, it get clot, but until unless it is filled inside our body, it did not get clot, right? So how it's possible? There is a hormone called heparin. Heparin remains dissolved with our blood. Heparin do not allow blood to clot inside our body. That's why heparin is known as the anticoagulant. Coagulation means clotting. Coagulation means clotting. So this is called anticoagulant or anti-clotting agent. Heparin is known as anti-clotting agent or anticoagulating. Got it? What is the role of heparin?
Yes, sir. Yes, sir.
So keep all these things in mind. If you want, you can take the screenshot of this one. If you want, you can leave this. I'll
Yes, sir.
dictate you. Yeah, I
Okay.
have to dictate it because until unless I won't dictate you, you will forget after 2 days, right?
Okay, sir.
So that's why dictation is important. I know. Today you will
Yes, sir.
understand. Today you will come to know that this is so easy, so good, right? And after four
Yes,
or
sir.
5 days, when you will get indulged with other chapter or other topic, You will forget. Half of the thing will washed out from your mind. That is a human tendency. Even it happens with me as well.
Yes, sir. Memory.
Yeah,
Short-term memory, long-term.
exactly. We are not supercomputer, or we are just human beings. Sometimes even I do forget. Here in the last slide, Shidika, we have studied about the component, right? So now we'll discuss with the purpose. I told you that clotting purpose to teach you clotting was that to make you understood the component of the blood. Here now we will discuss each and everything one by one. What is RBC, WBC, platelet, and all that. Got it?
Yes,
So
sir.
next part is RBC, red blood corpuscle. Look, uh, yes, formed element in NCRT. We are following the same sequence of NCRT. So why we call them capsules, not cells? My first question is, we had discussed, we had discussed already. Tell me why. Sometimes in the some books, some notes, they say this is red blood cells. They frequently say the red blood cells. That is a wrong notion, and that is the wrong concept. We cannot call it cell. We call it capsules, and they are also known as the erythrocytes. Tell me, Shirika, do you have any idea?
No, sir. I forgot.
Look, when I was teaching you cell, I said to you that to become a cell, there are certain criteria. It should have nucleus, protoplasm, and cellular component like mitochondria and all. RBC do not have nucleus, and many of the cellular component are missing. So first thing, they do not have nucleus, and the cell component organelle of the cell, they are missing. So because of these missing this missing thing, we do not call it cell. We call it capsules. So there is a no nucleus, there is a no cell organelle. We just call it capsule. Uh, their number in our body is 4.5 to 5 million per ml, right, or mm cubic. So that is their number. When I say 4.5 to 5, it means 4.5 in female and 5 in males. Abnormal, these are not in NCRT, but they are medical part. Abnormal rise of the uh, RBC is called polycythemia, and abnormal decrease of RBC is called erythrocytopenia. What is? So
Yes, sir.
polycythemia or erythrocytopenia. Okay. Size of RBC is 7.8 micrometer. 100 ml of blood contain 12 to 16 gram of hemoglobin. Right? Formation of RBC is called erythropoiesis. Right? Erythropoiesis. These are the term. Look,
Yes, sir.
whatever the line, the star is over there line. Like these all are the question already asked in medical exams. These are the question. The star point. Right? Lifespan is 120 days. Graveyard of RBC is called spleen. Look, so these are the concepts which you need to elaborate. So why spleen is called graveyard of the RBC? Tell me.
Because that's where the dead red blood cells are taken and um dispersed at.
Okay, very good. Look, spleen is called graveyard of RBC, and spleen is also known as the blood bank. Why? After 120 days, RBC die, right? So every day, number of the RBC dies, right? So when these RBC die after 120 days, the dead RBC they reaches to the spleen. So blood goes through the spleen. The spleen filter the blood and capture the dead one and let the live RBC let the go from it. So after the death, all the RBC reaches to spleen. The spleen break down the RBCs, right? That's why it is called graveyard of RBC. Suppose that the extra or more blood formation taken place in our body, right? So our body required only 5.5 liter, but more than 5.5 liter blood is there, right? So what will happen? Extra RBC will be stored in the spleen. That's why it is also known as the blood bank. So it is graveyard as well as blood bank, right? Anemia is the deficiency of blood. That stage is called anemia. Is it clear?
Yes, sir. Yes, sir.
One very interesting thing, RBC is the cell in our body which do not perform aerobic respiration. It performs anaerobic respiration. Got it?
Yes, sir.
This is the picture of RBC. There are these molecules are known as the hemoglobin. Hemoglobin molecule, they capture the oxygen. So it we represent with capital H, small b, and plus O2, HbO2. This is called oxyhemoglobin. Hemoglobin is made up of globin protein plus iron, that is called heme group, right? Formation of RBC takes place in the red bone marrow. Okay. So this was all about the RBC. And you do not have NCRT as well, Sridhika?
I have it, sir. I have it.
You have it. Okay. So keep on going with your NCRT as well, and keep on
Yes,
asking.
sir.
Right,
Okay,
erythrocyte.
sir.
It is there, 279 page number.
Okay, sir. 1 second. I'll just read to it.
Yeah, take your time.
It's clear, sir. Everything is clear.
Okay, great. Now come to the second one, that is a WBC, or we call that erythrocyte, leukocyte. This is very important and interesting too. Look, uh, first thing, WBC are all known as leukocyte, right? Uh, they have nucleus, fine. So they have nucleus and some cell organelle are present. So their size is 10 to 20 micrometer, and they have nucleus and cell organelle. Formation of WBC is called leukopoiesis. Fine. So, like we have studied that formation of RBC is called erythropoiesis. So, formation of WBC is called leukopoiesis. There is a no fixed shape, right? Or they have the amoeboid shape. So, what happened? These RBC they keep on flowing with the blood, WBC, and there are some space or cracks in our blood vessels. So they leak out, they change their shape and they leak out from the blood vessels, and they keep on roaming here and there along with the uh, blood, right? Lymph or plasma, blood plasma or lymph, right? And they kill the germs over there. That is the benefit of leaking out. Fine. Their number are 6,000 to 10,000 per mm cubic. The ratio of WBC and RBC is 1 is to 600. This is important. Rise of WBC, that is called leukocytosis. Abnormal rise of WBC is called leukemia or blood cancer. Do you know what is the blood cancer? What happened in the blood cancer? Any idea, Shidika?
The blood cells get nothing.
Look, the blood cell start eating their own cell.
Yes, sir.
Abnormal increase of the WBC takes place. So, what is the function of WBC? The function of WBC to kill and eat the germs. Rather than eating germs, it start eating our own cell, body cells, right? That's why means technically we call that autophagy, right? So that is uh, that is blood cancer. That's why you know that blood transfusion is required for the blood cancer patient, right? So abnormal rise of WBC is called leukemia or blood cancer. Now, look, WBC, they are called leukocytes. There are two type of the WBC, a granulocyte and granulocyte. A granulocyte, they do not have granules, and granulocyte, they have granules. Means in what kind of granule? The granules in their protoplasm. Further, a granulocyte, right? This notes is very important that I have made by myself, and it is very important to explain everything about a WBC. Fine. So that you need to cram. In fact, after taking the screenshot, you need to cram, Siddhika. So WBC is of or leukocyte is of two types: a granulocyte and granulocyte. A granulocyte are those which do not have granules in their protoplasm. They are further of two types: lymphocyte and monocyte. Lymphocyte further may be B lymphocyte and T lymphocyte. Try to understand. Lymphocyte may be B lymphocyte and T lymphocyte. B lymphocyte or B cells. So normally B lymphocyte called B cells. They are they produces antibody and they develop in liver. T cells, they develop or mature in thymus gland and they directly attack the germs, right? So that is a B cell and T cell. They are non-motile and non-phagocytic. Non-motile means they cannot roam from here to there, right? And they are non-phagocytic means they do not eat germs or bacteria. Rather, they kill them with the help of production of antibodies. They produces antibody and destroy microbes and toxic and tumor cells. That is important. Our body have self-healing capability. Our body have self-defense capability. These lymphocytes can naturally kill the cancer cells or tumor cells. So they are 20 to 25% of WBC every blood sample, right? Or their lifespan is for few days. Now come to the monocyte. Monocyte is largest of all WBC. They are motile and phagocytic. Phagocytic means they eat the germs and bacteria. They engulf the bacteria and cell debris. Their lifespan is 10 to 20 hours. So these were agranulocytes. Let's talk about the granulocytes. So granulocytes are of three types. So normally they have granules. They're of uh, their lifespan is 4 to 5 days. They have granules. They're of three types: eosinophil, basophil, and neutrophil. Eosinophil, they they increase their number in allergy, asthma, and hay fever. They help in uh, dissolving the blood clot. Right? They are non-phagocytic, means they do not eat bacteria. They are similar like lysozyme, they destroy the protistans, right? So they are responsible for dissolving blood clotting, that you need to keep in your mind. Now come to the basophils. They release heparin, serotonin, and histamine, right? Heparin and serotonin, they are a hormone, and histamine, it's a type of protein, right? Which carry the allergy, which generate the allergy. And now come to the neutrophils. Neutrophils, they are the most numerous, means they are 60 to 65% of WBC. They eat harmful germs, and that's why they are phagocytic. Go through with it and ask me if you have any doubt in any point.
Okay. Just 2 minutes. Clear, sir.
Okay. Now come to the platelets. Then you go through with the NCRT. Come to the platelets. So platelet, they are simple. Their number is 1.5 lakh to 3.5 lakh per ml or mm cubic. Nowadays, all of us, we know the platelet count, and everybody talk about the platelet count, and the platelet count is going down or coming up, improving and all that. Increase in platelet is called thrombocytosis. Decrease is called thrombocytopenia. They are colorless. They do not have nucleus, and they are means they are non-nucleated. Their lifespan is 1 week, and formation takes place in megakaryocyte cells of the bone marrow. Special kind of the cell they produce. Now go through with the formed element of the blood or component of the blood, and ask me if you have any doubt. Then we'll move to the next topic, that is blood group. Go through.
Okay, so just a few minutes. Clear, sir.
Blood group. Look, the A, B, and O blood group. They were discovered by Steiner in 1900. So Steiner was a scientist who discovered this blood group, and three blood group he discovered. After that, the AB blood group was discovered. It was discovered by Di Castello and Stanley. Right? The name of the blood group is given on the basis of antigen. Now tell me what is the antigen or what are the antigen? Define antigen and antibody. Shidika, tell me.
Uh, Antigens they're present on our red blood cells, the antigens and antibodies. The antibodies are released by the antigen.
Look, the definition means antigen is not fixed for every person. Means they are not same for every person. Look, Shidika, what are antigens? So basically, anything which goes inside our body, when it reaches to our blood, if our blood start producing WBC or they start reacting against it, that is antigen for it. Let's say you can say in this way, if any biological or non-biological thing, if it enters inside body and our immune system start working against it or it get activated, then this is antigen for us. Let's say, for example, suppose that I am allergic to dust, but you are not, Siddhika, allergic to dust. So if I'm allergic to dust, whenever dust is there, you are normal, you are behaving normal, you hardly have any trouble. But I start sneezing, right? After some time, I will get fever, right? I mean, why? Because that dust is antigen for us. When it goes inside our body, our antibodies formation start take place in our body. that is antigen for me, but not for you. In the same way, suppose that you have allergy with the pollen grain. You cannot go for the morning walk. When you reaches to a garden at the morning time, what happened? You start sneezing and then you feel the fever and all that. It means pollen grain, they are working as antigen for you, but they are normal to me, right? So look, in this case, we can say that antigen can be different for different person. But normally, or in the nutshell, we can say anything which is biological or non-biological, when it enter inside our body and our body start producing antibodies against it, then this is antigen, or our immune system get activated, then it is antigen. Look, antigen can be biological or non-biological. In biological antigen, we can see that that can be bacteria, virus, protozoa, fungus, and pollen. Non-bacterial antigen, non-biological antigen, they can be smoke, dust, and chemical. So that's how we say it antigen. Now, what are the antibody? Whenever suppose that let's say the bacteria is antigen. Now, let's take the example. So bacteria, let's consider bacteria as antigen. So whenever bacteria will go inside our body, there are two type of the defense mechanism, or there are two method of defense mechanism. Like one method, our WBC, as soon as WBC will get to know that bacteria is there, they will go to there and they will eat the bacteria and die themselves. This process is called phagocytosis, cell eating, right? So that's how they defend our body. They eat the bacteria, they die themselves. They are the soldier of our body. In second process, whenever bacteria reaches to the WBC, WBC starts secreting a special type of protein in the blood that is called antibody. And these protein remains dissolved in the blood for years. So whenever the bacteria will come in the contact with antibody, the bacteria will die. So they are non-phagocytic, means they do not eat directly, but they kill the bacteria. Both are killing the bacteria. One is phagocytic, one is non-phagocytic, and both have different mechanism. But uh, the thing which is produced by this one, this is called antibody. Tell me, is that clear?
Yes, sir.
Now come to the blood group. So antigen antibody perfectly clear. Kirika.
Sorry, sir.
So they are perfectly clear to you.
Yes, sir.
Fine. Keep it in mind when I will teach you the vaccine and all that. You need to recall it, right? Keep it in mind. Fine. This is very important concept. Now come to the blood group. So I said to you, ABO was discovered by Ladistino in 1900. Remember the name of the scientists because they are good. They are important for your one marker question. AB was discovered by De Castello and Steny, means someone else discovered. The name of the blood group A, B, AB, and O has been given on the basis of antigen found on their surface. A blood group have A antigen, B blood group have B antigen, AB have AB, and O group, it do not have any antigen. That's why it has called O or zero or neutral. In contrast, the blood group A have A antigen, but the antibody which is produces B. The blood group B have B antigen, by antibody it produces A. AB blood group do not produce any antibody. That's why this is universal recipient. It can take the blood from anyone else, any blood group. Sorry. O blood group, it do not have any antigen, but it have AB antibody. That's why it can be given to anyone. That's why this is called universal donor. Take the screenshot and let me know if you have any doubt. And then go to the blood group, ABO grouping.
Clear, sir.
Clear? Okay. So I have a question, Shadika. The blood group of my father was O, and the blood group of my mother is A. What is the probability of blood groups which should be present in my brothers? Can you tell me? What is the probability of blood group in my brothers?
Uh,
Any idea?
A,
A.
because I know O because O is the most common
A.
one.
Okay. So should it be A O both, or only A will be there?
For just a single person or multiple?
Multiple person. Like we are 8 brothers.
Uh, you have 8 brothers?
Yeah, we are 8 brothers,
Oh, wow.
and we are living in same home.
That's so impressive, sir.
And we share the same kitchen. Like
That is so impressive, sir. 8 brothers.
yeah,
What's the
and
thing?
I am the sixth one.
Oh, so you're the younger one.
Yeah, I am the younger one. I am the sixth one. I have the two more younger brothers.
Oh, you don't have to stay?
Yeah.
You don't have to stay?
No, we don't. Unfortunately, I don't. We don't have sister, and we are happy that
Oh,
I have uh, one small daughter, and 10 month old,
oh,
and I have two nieces as well. So now we are fortunate enough in our generation. I do not have any sister.
oh.
Even my like maternal uncle, they do not have any daughter. So can you tell me the probability of the blood group in
uh,
my brothers? What should be the probability?
the probability would
What are the option for my blood group? Tell me. What should be the option for my blood group? Means, can you guess what should be the my blood group?
-8, -B, negative.
How B possible?
Sorry, positive A,
Right. Very good.
and negative O, positive O, AB.
AB is not
No.
possible. Look, that's what I want to train you. Look, how you can tell. I'll tell you what is the what is the negative or positive. We'll talk about that. First, talk about the blood
Yes,
group.
sir.
Let's talk about the blood group. Suppose that I said to you that my father had blood group O, right? So basically, his blood group was O negative. Means we have two sets of chromosome, right? So this is true. This is fixed, right? His blood group will be IO and IO. That's if someone have positive or negative. Forget about this positive or negative. Just O blood group. My mother have my mother has blood group A, right? So my father have O blood group. So there is only one option. The O can be this is recessive, so O can be always in this state. My mother have A blood group. It means that either the genes of my mother will be IA, IO. Why? A is dominant, right? We'll discuss dominant and recessive. Have you studied it in 10th class? 10th standard it was
Yes,
there.
sir.
Okay,
Yes,
fine.
sir. That's okay.
So O is recessive. So O will always show itself in homozygous condition. Homozygous, when both genes are similar, we call it homozygous. I will let me recall you. When both genes are different, we call them heterozygous. Clear? So O blood group
Yes,
will
sir.
be always there in homozygous state, right? Because O is recessive. Here A is dominant and O is recessive. So dominant will show itself to IA, and so this we call heterozygous A, and This is homozygous A. Is it clear? Is that clear?
Yes, sir. Yes,
Okay.
sir.
So first make the first combination. I said my father have O blood group, and let's take my mother's blood group is this one, IAIO, right? So IAIO. So now what is the probability? So how we will make the probability? We will make a cross. We'll make cross with IOIA. So what can be the blood group? Combination is GIOIA. So what will be the blood group? Silika? A, because A is dominant.
A.
A, right?
Yes, sir.
Now we will pick this one and we will cross with this one. Now when we multiply, what should be? IO.
IO.
What should be the blood
O
group?
blood group.
O
O.
blood group. Very good. Now what we will do? We will take this one now and we will multiply with this one first. So what is the combination? IO.
IA.
What should be the blood group?
A blood group.
Very good. Now let's multiply this with this one. So that is the IO and IO. What's
IO.
your blood group? O.
O.
So if my mother is heterozygous A, right? If mother is heterozygous A,
Yes.
in this situation, first situation, so 50% of my brother will have probability A blood group and 50% chances to be what? O blood group.
O
Clear?
blood group. Yes, sir.
You can tell me what should be. So none of us will have B blood group or AB blood group, right?
Yes, sir.
Let's talk about that. My father is O, so his genotype will be C, right? Similar. But my mother is homozygous A. We have IA. Now what should be in this situation? IA and IO. What should be the blood group?
A. A again.
Right. A again. So IO and IA.
All will be A.
Exactly. All will be A. Right. So there are two options. Your answer should be that either you will have A blood group or you will have O.
O.
Right. There is no other option. Got
Yes,
it?
sir.
How you can
Yes,
calculate
sir.
the blood group?
Yes, sir.
So this was this with the help of this example, I just shown you that what should be the blood group, and after that you can calculate the even you can tell the you can guess the blood group of your like sister and brothers. Got it? With
Oh,
the
okay,
help of
sir.
you, yeah, with the help of your. So here should be 100% A. Look
Okay, sir. Got it.
now let's conclude. Look, take one more logic. My blood group A positive, right?
Yes, sir.
And my elder brother have blood group O positive. Got it?
Yes,
So now
sir.
guess what should be the genotype of my mother?
A.
Which A? IAIO or IAIA?
IAIO.
Exactly. That's how I can tell that my mother is heterozygous, not homozygous.
Yes, sir.
Because one of my brother is O, like I have O brother, it means this combination is there. And this combination is not there.
Yes,
Right?
sir.
So she is a heterozygous A and my father is homozygous O. Got
Yes,
it?
sir.
That's
Yes,
how I
sir.
tell. Now do this experiment with someone else, right? You will come to know. That's how this is
Okay, sir.
important. This combination, this it will be required for you in 12th class in genetics, heredity and variation. That's why I told you. Now we will talk about what is the positive and negative blood. So I think it should be clear to you. This one, crystal clear.
Yes, sir. It's clear.
Fine, great. Come to here. What is the first? Let me tell you what is the positive and negative blood. There is nothing benefit or demerit of positive and negative blood group. Shidika, if you have, suppose that I have A positive and you have A negative. So neither I have any advantage of this positive, A positive means positive blood group, or nor you have any disadvantage of negative. Right? First thing. The second thing, what is the positive and negative? So every RBC have a covering of protein around it. This protein is called RH protein or RH factor. It had been discovered in a monkey called rhesus monkey. So that's why we call it RH protein. RH means rhesus, right? So this protein had been discovered in rhesus monkey. It was discovered by Ladsteiner, right? And Wallner. Fine. So Ladsteiner and Warner discovered this blood group, right? Positive and negative blood group. RH protein. So if RH protein is present around the RBC, we will call the person have positive blood group or RH positive. If RH protein is absent, you will say that person have negative blood group. Got it? What is the A positive, A negative, or what is O positive, O negative? Is that clear, Shadika?
Yes, sir, it's clear.
Okay. Now, what will happen if a wrong blood group is given to any person? Suppose that a person have A blood group and unfortunately someone have given B blood group. What will happen with that?
Uh, the blood group will be attacked.
Blood group will?
The other blood that is transfused, it will be attacked by.
Okay. Means it will be attacked by the body blood group.
Yes, sir.
Okay. Means the recipient. Yeah. Almost you are close to the answer. So definitely, suppose that I have a blood group, his B cells will come in my body. So my body will it will treat B blood group as an antigen. Got it?
Yes, sir.
So you're right. That will happen. And what will happen due to this? Due to the wrong blood group transfusion, suppose that A is given to the B, the RBC will means different A and B blood group RBC, they will clump like this way. They will clump like
They will.
right? Clump.
Okay.
They will get adhere with each other. We call it clumping, right? And this
Yes, sir.
reaction is called agglutinization. I have written over here. Agglutinization. I will dictate in next class.
Okay, sir.
So what will be your answer? Because of the wrong blood group, or if we give the wrong blood group a person, the blood clumping or agglutinization will take place, and because of this, a person will die. Because why? If the blood clumping will take place, the RBC will clump down, they won't carry the oxygen anymore, and the person will die because of the stoppage of supply of oxygen. Is it clear?
Yes, sir, it's clear.
Now, what is lymph? And how does the lymph formation take place? Right. Fine. So, what is lymph? Try to understand, Siddhi. So, these are the blood vessels or blood capillaries. Blood is flowing in these vessels, right? Now, I have told you in the tissue classes when we were discussing, we have discussed lymph in short. So again, we are discussing the same thing. Do you remember? We have discussed that lymph is a middleman. Remember, Siddhika?
Yes, sir.
Okay, the same thing is here. So these are the blood vessels, RBC, and rest of the blood is flowing over there, right?
Sir?
Yeah.
I actually have guests coming over.
Okay, fine.
And the class early.
Yeah, sure.